So KRAS G12D is the most common KRAS mutation. Accounts for about 40% of pancreas cancer and 15% of colorectal and 5% of non-squamous non-small cell lung cancer. Unlike the G12C inhibitor, there's no real approved treatment option at the moment for G12D. So ASP3082 is a highly selective, novel, first-in-class KRAS G12D targeted protein degrader. It works by degradation, but it has a three component. The molecule has three components of a KRAS binding component and E3 ligase binding component, and there's a linker in between them. Once ASP3082 engages into the protein of interest here, KRAS G12D, it will call for the E3 ligase to its proximity, and it will start the ubiquitination. Therefore, by tagging the protein, as this continues, the protein will be tagged as disposable protein. Therefore, the proteasome intracellularly will degrade the protein and will dissolve. So growth inhibition occurs. So that's the mechanism of action. It's highly novel. They've tested it in multiple different molecules, and this was the best in class. We're able to move into the actual clinic.
Sorry, it's been a very long time.
Sure, so that's where really the biggest interest is. I mean, this is the first in class, first in human, and first KRAS G12D specific targeted therapy we're presenting. So there's a lot of excitement for sure. We're able to achieve really good responses in an advanced setting. These were patients who had pancreas cancer in a third line setting, and plus. So 93% of the patients received this in a third line setting and plus, meaning they didn't really have any other treatment options other than new clinical trials. We are able to achieve good response rate, encouraging response rate, which is not really expected in that setting without chemotherapy. So 18.5%, 5 responses were seen, and if you restrict it down to third line and below, it was a 23.5%. Can we increase the percentage by moving the treatment further into the earlier line of the treatment? I think so. I think it's precedent in a lot of different data in different clinical trials ongoing show very similar example that as you move along into the earlier setting, when the disease is not as aggressive and it's not as burdensome, you'll get more effect. So eventually we want to go there. Whether we're going to do it with the monotherapy or combination is to be determined. At the moment we're expanding and also evaluating the different lines of treatment and in monotherapy. It seems to be very safe, so there's no question about combinability. It's rather of which setting we're going to go. Is it going to be combined with something or monotherapy? That's something we're still waiting to have some more evaluation for.